Hello everyone, I'm Margin. It's my great pleasure to be here to share some knowledge about nucleotide. So in this chapter, we are going to discuss DNA, RNA, and the flow of genetic information. So till now, there are more than 960 bacterial genomes were sequenced. Bacterial genomes are circular and the genes are densely packed. Their genome size are around 2 to 10 million base pairs, and also there are around 470 to 70 million genes in different strains of bacteria. There are more than 200 eukaryotes genomes were sequenced. Eukaryotes genomes are linearized. On average, half of the gene functions are known. There are less than 40,000 genes in human genome, while they code for more than 120,000 proteins. Most importantly, 98% of human DNA is non-coding. They did not make proteins. Around 3% of human DNA are simple repeats. We call them stagnates or macrostagnates. So beyond that, half of the DNA are mobile elements. We call them DNA transposons or retrotransposon. The Human Genome Project was an international scientific research project with the goal of determining the base pairs that make up human DNA, and of identifying and mapping all of the genes of the human genome from both a physical and a functional standpoint. It remains the world's largest collaborative biological project. The idea of Human Genome Project was packed up in 1984 by the US government. The project formally launched in 1990s and was declared complete on April 14, 2003. Increasing our understanding of Earth's biodiversity and responsibly stewarding its resources are among the most crucial scientific and social challenges of the new millennium. These challenges require fundamental new knowledge of the organization, evolution, functions, and interactions among millions of the planet's organisms. Herein, Earth's Biogenome Project, a moonshot for biology that aims to sequence, catalog, and characterize the genomes of all of the Earth's eukaryotic biodiversity over a period of 10 years. The outcome of the EBP will inform a broad range of major issues facing humanity, such as impact of climate change on biodiversity, the conservation of endangered species and ecosystem, and the preservation and enhancement of ecosystem service. Since we talk about the genome, let's look back at the cell biology. Prokaryotes are organisms whose cells do not contain a distinct nucleus. Eukaryotes are organisms whose cells have a nucleus enclosed within the membranes. This fundamental difference at a cellular level suggests that these genomes are different. In the next few slides, we will mainly focus on the genome of eukaryotes. The sugar in deoxyribonucleic acid is deoxyribose. Position 2 carbon atom of the sugar lacks the oxygen atoms. That is linked to the 2 carbon atom of the ribose, while the RNA has oxygen atoms at the position 2 carbon. This figure is the X-ray photograph of the DNA. Based on this, James Watson and Francis Crick created the first accurate model of DNA structure in 19 P53. DNA is a double helix. This makes DNA look like a twisted ladder. Each molecule of DNA is composed of a backbone and the outside and the bases that point towards the middle of the molecules. The monomer of DNA is the nucleotide. DNA is built following the complementary base pair rules. This states that A always pairs with T, T always pairs with A, C always pairs with G, G always pairs with C. While in RNA, adenine pairs with the U. Nuclear organization refers to the spatial distribution of chromatin within a cell nucleus. At a slightly larger scale, DNA looping or DNA fiber can physically bring together DNA elements that would otherwise be separated by a large distance. At the smallest scale, DNA is packed into a unit called nucleosomes. Double-stranded DNA wraps around the histone proteins to form nucleosomes. The nucleosomes are caught into a 30-nanometer chromatin fiber. When the cell undergoes mitosis, the duplicated chromosomes can dense even further. The nucleosome's core particle consists of approximately 146 base pairs of DNA. The nucleosome consists of 2 copies of each of the core histones, H2A, H2B, H3, and H4. Core particles are connected by stretches of linked DNAs, which can be up to about 80 base pair long. Different species have different chromosome count. Tomatoes have 24 chromosomes, but potatoes have 48 chromosomes. For humans, we have 46 chromosomes. Chromosomes contains a linear array of genes, and each gene resides at the particular location on the chromosome. The location is more formally called a genetic locus. The alleles of a gene are the different forms that are found at its locus. Although generally there are up to 2 alleles per locus in diploid individuals, a population might have many alleles of a single gene. Each gene is a part of a continuous sequence of the DNA. The gene structure and the gene expression matched mechanism in eukaryotes are far more complicated than in prokaryotes. In typical eukaryotes, the region of the DNA coding for a protein is usually not continuous. This region is composed of alternating stretches of exons and introns. A typical multi-exon gene has the following structure as illustrated in the top panel of this slide. It starts with the promoter region, which is followed by a transcribed but non-coding region called 5' untranslated region. Then follows the initial exons, which contains the start codon. Following the initial exons, there is an alternating series of introns and interlo exons, followed by the terminating exons, which contains the stop codon. It is followed by another non-coding region called the 3' untranslated region. Ending the eukaryote genes, there is poly-A signal, the nucleotides adenine repeating several times. As the bottom panel shows, genes are conserved in different species because of the evolution, like very famous tumor suppressor genes P53. It conserved among mouse, rat, and even in small dinosaur fish, but they have different composition of exons and introns. In human genome, Genes are sparsely located in the chromosomes. Genes are interrupted by repeat DNA sequence, which is labeled as blue in this figure. As shown in the pile B of the figure, genes of bacteria are very close to each other. They don't have so many DNA repeats sequence. As shown in pile C, it is the same phenomena in Drosophila chromosomes, but in males. There are more DNA repeat sequences than genes, which is striking. This discovery nominated a Nobel Prize novelty. Transposable elements, also known as jumping genes, are DNA sequences that move from one location on the genome to another location. These elements were first identified by geneticists Barbara McClintock of the Cold Spring Harbor Laboratory in New York. Biologists were initial scratchers of McClintock's discovery. Over the next several decades, however, it's apparent that not only do TEs jump, but they are also found in almost all organisms, both prokaryotes and eukaryotes, and typically in large numbers. Barbara McClintock was awarded as Nobel Prize in 1983 for her discovery of mobile genetic elements. There are two methods of transposition: cut and paste mechanism, or copy and paste mechanism. Structure of DNA transposons. Two inverted tandem repeats flank the transposase genes. Two short tandem site duplications are present on both sides of the insert. Two transposes recognize and bind to TIR sequence, join together and promote DNA double-strand cleavage. The DNA transposase complex then inserts its DNA cargo at a specific DNA motif, as well as in the genome, creating short TSDs upon integrations. After talking about the gene structure and the genome structure, can we manipulate genomes as what we want to? The answer is yes. I will use CRISPR to briefly introduce the hot area of gene editing. CRISPR was identified an unexpected defense mechanism that is bacteria use for fighting off virus in 2007. A buzz announcement came in 2012, followed by crucial first step in 2013. Now, it has matured into a molecular marvel, and much of the world, not just biologists, is taking notice of the genome editing method CRISPR. CRISPR was awarded as Breakthrough of the Year in Science in 2015. You can edit genes in almost all organisms by CRISPR technique. Basically, researchers create a small piece of RNA with a short guide sequence that attaches to the specific target sequence of DNA in the genome. The RNA also binds to the Cas9 enzyme. Once the DNA is cut, researchers use the cell's own DNA repair machinery to add or delete pieces of the genetic material, or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. Here is some reference, and thank you for your attention.